I'm Aki. I am the CEO and co-founder of Elixogen Therapeutics. We are a clinical stage biotech company located in Baltimore, Maryland. And our lead clinical candidate is an autologous cell therapy for telomere biology disorders with bone marrow failure, and it's called EXG34217. Telomeres are the protective end caps of your DNA that are present on all of your chromosomes, and they naturally shorten over time as we age. And people with telomere biology disorders are born with short telomeres and have accelerated shortening due to a mutation in their telomere maintenance genes. And when the telomeres in their various cells get too short, various issues start to occur. So one of the most common is a progressive dysfunction in the person's ability to create new blood cells. And this is something that normally takes place in the bone marrow thanks to hematopoietic stem cells, or HSCs, which are the bone marrow stem cells. But when the HSC telomeres get too short, they get worse and worse at producing new blood cells, which includes immune-related cells too. So TBDs are typically diagnosed from either issues with blood count, but also genetic testing. Telomere biology disorders are very complicated and can manifest in many different ways, but the most common and also potentially the worst part is their slow inability to create blood cells. Their reduction in their hematopoiesis, which lowers their blood count. Currently, there are some mitigations with some trade-offs, but the ultimate treatment is a hematopoietic stem cell transplant in which a donor's HSCs are given to the patient. However, the donors need to be matched, which is one hurdle. Preconditioning may need to be done, which includes chemo or radiation therapy, and immunosuppression might also need to be continued after the transplant. Some additional risks and hurdles are due to the fact that the cells being transplanted are someone else's. EXG34217, our lead clinical candidate for telomere biology disorders, is an autologous cell therapy, which means that your own hematopoietic stem cells are treated and given back to you. For the therapy, the HSCs are first mobilized from the patient's bone marrow into their bloodstream, and then they're isolated through a process called apheresis. Next, the HSCs are treated with a special gene called ZSCAN4, which extends telomeres. And then finally, the HSCs are infused back into the patient with the extended telomeres. And all this takes place with an inpatient time of only roughly 28 hours. And the therapy addresses the root cause of the telomere biology disorders, the short telomeres, in the cells that need it the most, the HSCs, with the goal of maintaining and potentially even restoring the patient's production of their blood cells. We're currently in phase 1/2 clinical trials at Cincinnati Children's Hospital, and we have presented preliminary data before, but are planning to report additional interim results in the very near future. What was presented at ASH 2022 was a case study for one patient where we showed that the telomeres of the HSCs going back to the patient were already extended by ZSCAN4 for the HSCs. And then on top of that, at the 6-month time point, there was an emerging population of cells that were potentially produced from those HSCs that also had extended telomeres, some of them even reaching the normal range. And so that is very exciting preliminary data. Some of this is still in progress. And so with the current phase 1/2 clinical trials, what I can predict for now and what I can say is that additional interim results in the near future.